Hello. There are two things you're probably doing right now, or at least thinking about. Breathing and eating.
It's all to fuel one process, cellular respiration. Cellular respiration is a catabolic process that all living organisms have to undergo in order to generate energy for other cellular processes.
Every living organism on the planet goes through this process as it is essential to create the currency of energy called ATP.
The equation for cellular respiration is as follows. The reactants are a simple sugar, usually glucose, and 6 molecules of oxygen.
The products of cellular respiration include 6 molecules of carbon dioxide, 6 molecules of water, and energy in the form of ATP molecules, usually about 32 to 38 molecules.
Cellular respiration, however, isn't a simple process. Instead, it has a few steps. The first is glycolysis. This is followed by pyruvate oxidation. Next comes the citric acid cycle, and then the electron transport chain.
The purpose of the first 3 steps of cellular respiration is to generate things called electron carriers, known as NADH or FADH2. These electron carriers fuel the electron transport chain by donating their carried electrons in the final step of cellular respiration.
The following is a breakdown of the mechanisms involved in each of the first 3 steps.
Glycolysis. The goal of glycolysis is to break down the 6-carbon sugar into 2 3-carbon sugars called pyruvate molecules. It's a 10-step process that initially requires the input of 2 ATP molecules to destabilize the glucose in order to overcome the activation barrier needed to start the rest of the reactions.
The 2 ATP molecules are used to phosphorylate the glucose, which forms a 6-carbon fructose 1,6 biphosphate. This is then cleaved into 2 3-carbon sugars called glyceraldehyde 3-phosphate, or G3P for short. The G3P then undergoes a series of redox reactions.
It is oxidized and releases electrons in order to reduce these electron carriers.
This diagram shows not only the 2 ATP molecules invested at the start of glycolysis, but the cleavage of the 6-carbon sugar into the 2 3-carbon sugars. Finally, it shows how these 3-carbon sugars are oxidized in order to generate energy.
This process generates NADH from NAD+ and FADH2 from FAD+. In addition, the whole process of glycolysis produces 4 ATP molecules, but since 2 molecules were used in the beginning, the net gain of ATP is 2. The final products of glycolysis are
2 molecules of NADH, 2 molecules of ATP when we take into account that the net gain is only 2, and 2 molecules of pyruvate, which will move on to the rest of the steps of cellular respiration. The next step in cellular respiration is pyruvate oxidation.
The purpose of pyruvate oxidation is to transform the 2 molecules of pyruvate obtained in glycolysis into 2 molecules of acetyl-CoA.
This process reduces 2 NAD+ molecules into 2 molecules of NADH. In addition, for each molecule of pyruvate, 1 molecule of CO2 and 1 molecule of NADH are created. However, because the process has to happen twice, once for each pyruvate, the final products of pyruvate oxidation are
2 molecules of NADH, 2 molecules of CO2, and 2 acetyl-CoAs.
This diagram shows the molecule of pyruvate moving into the mitochondria, where it loses 1 carbon molecule to form CO2, and where it reduces NAD+ to NADH. It also shows the addition of coenzyme A, forming acetyl-CoA.
The next step of cellular respiration is called the citric acid cycle. This cycle takes 2 molecules of acetyl-CoA and puts them through an 8-step process located in the mitochondrial matrix.
The purpose of the citric acid cycle is to reduce electron carriers, which can then be used to fuel ATP synthesis using the electron transport chain. At the start of the citric acid cycle, one molecule of acetyl-CoA combines with one 4-carbon molecule of oxaloacetate. This forms one 6-carbon molecule of citrate.
This citrate molecule is then oxidized through this 8-step process, becomes a 5-carbon sugar and then becomes a 4-carbon sugar to actually give you the final product of oxaloacetate.
This diagram shows the regeneration of the oxaloacetate through the 8 steps of the citric acid cycle. It also shows how during these 8 steps the electron carriers are reduced. Finally, it shows us the products of the citric acid cycle. For each turn of the citric acid cycle, and there are 2, we produce 3 molecules of NADH, 1 molecule of GTP, and 1 molecule of FADH2. In addition, we release 2 molecules of CO2.
Final step in cellular respiration is the electron transport chain and the synthesis of ATP. The first 3 steps of cellular respiration generate only 2 molecules of ATP, though this is the molecule most useful to the cell to be used as energy. In order to supplement and create more ATP molecules from the breakdown of simple sugars, the cell uses an electron transport chain fueled by the electron carriers NADH and FADH2. This works as follows. The NADH and FADH2 generated in the earlier steps of cellular respiration, glycolysis, pyruvate oxidation, and the citric acid cycle, travel to the inner mitochondrial membrane. The first 3 steps of cellular respiration generate only 2 molecules of ATP, though this is the molecule most useful to the cell to be used as energy. In order to supplement and create more ATP molecules from the breakdown of simple sugars, the cell uses an electron transport chain fueled by the electron carriers NADH and FADH2. This works as follows. The NADH and FADH2 generated in the earlier steps of cellular respiration, glycolysis, pyruvate oxidation, and the citric acid cycle, travel to the inner mitochondrial membrane. There, NADH and FADH2 are oxidized, releasing 1 hydrogen plus ion from each, which is then pumped through a channel protein and out into the mitochondrial matrix between the inner and outer mitochondrial membranes. This establishes a proton gradient between the inner mitochondrial membrane and outer mitochondrial membrane. When NADH and FADH2 are oxidized, they also release 1 electron each. The electrons travel through a series of integral membrane proteins, called the respiratory chain. Within the inner mitochondrial membrane, at the end of the respiratory chain, the electrons are given to oxygen, forming water molecules. This is why in the formula for cellular respiration, water is produced. Once a proton gradient has been established, with a higher concentration of hydrogen protons outside the mitochondrial membrane, ATP synthase uses the proton gradient to generate energy for phosphorylation of ADP into ATP. This is called chemiosmosis, or oxidative phosphorylation. ATP synthesis pumps the hydrogen ions back through the inner mitochondrial membrane to harvest their energy. This combines ADP with inorganic phosphorus, forming ATP.
As a result of oxidative phosphorylation, the cell is able to generate somewhere between 32 and 38 molecules of ATP for each molecule of glucose that undergoes cellular respiration. This gives it the energy it needs to fuel further chemical reactions and carry out other cellular processes. After all of these steps, the process of cellular respiration is finally over. The final products are 32 to 38 molecules of ATP, 2 molecules of GTP, 6 molecules of water, and 6 molecules of carbon dioxide. Thanks for watching. We hope you had fun.